So we concluded from that probably that there was more clearance, or, well, in the second point here, in Alzheimer's disease brain, much more viral DNA located in plaques, less in normals, possibly because less produced and or more cleared. But the point is also that in both Alzheimer's disease and nature normal brains, nearly all plaques contain viral DNA. In a fairly recently published paper, we refuted the possibility of artifactual co-localization, and there are some further points, but again, I have further arguments against that, but there's not really time to say that. But these data, together with the accumulation of A-beta in virus-infected cells and also in infected mouse brains, suggest the two have to be considered together, because an association doesn't prove causality. But the two together strongly suggest that the virus is a major cause of toxic A-beta products and plaques. Well, just to finish off with telling you a little bit about antiviral agent. This one is very much frequently, well, almost always used in cases of herpes simplex encephalitis. And I don't think there's time, is there?
For that, we do have time.
Okay. Well, I don't know that I need to read this out, but it's a nucleoside analog, and the point is it stops viral replication, and it depends on the presence of the viral thymidine kinase, which is obviously going to be there only if the virus or viral DNA, reactivated virus, is there in the cells. And it phosphorylates, the kinase phosphorylates the ACV to its monophosphate form, and then cell enzymes further phosphorylate it. And these triphosphate competes with G2P as a substrate for DNA polymerase, and so new DNA prevents elongation. So the point really is it specifically targets cells with virus in them and stops virus replication. I've got a scheme here which is what we think might be happening in the actual process and what we want to try to stop. The virus enters the brain, becomes latent. Stress, peripheral infection, and so on, immunosuppression could cause reactivation. This is a schematic showing reactivated virus particles in the cell, not to scale, obviously. The virus causes accumulation of A-beta by increasing secretase degradation of amyloid precursor protein. These are meant to be, these red bits are beta amyloid, which then aggregates to a certain extent, and this shows the cell before it actually lyses and disintegrates. And this is a diagram of an amyloid plaque showing viral DNA and amyloid within it. So the answers to our question we asked at first, is viral DNA present in normal brains? And the answer is yes in the elderly. I've got to say that it's present in only a low proportion, very low proportion of brains of younger people. We think it gets to the brain in older age because of the decline in the immune system. Second question, is the virus active in brain? And yes, perhaps recurrently. Is the virus in brain associated with Alzheimer's disease? Yes, in ApoE4 carriers. And is there a causal link with Alzheimer's disease and neuropathology? The answer is yes, with plaques and Alzheimer's disease like tau. So to summarize, the virus resides in and has replicated in the brain of many elderly people, and that, together with ApoE4 carriage, confers a strong risk of Alzheimer's, and incidentally, ApoE4 confers a strong risk of a number of cold sores, fever blisters. And I didn't have time to share this, but we found that vaccination of mice with a mixed viral glycoprotein prevents viral latency in brain, or reduces it very greatly, which is interesting. And the virus causes accumulation of A-beta and Alzheimer's disease like tau, and increases levels of the relevant enzymes. The viral DNA co-localizes with amyloid plaques in Alzheimer's disease brains, and acyclovir reduces virus-induced A-beta accumulation and Alzheimer's disease like tau phosphorylation in cell cultures, in infected cell cultures. And as a side issue, just to say that ApoE seems to determine severity of damage or susceptibility to infection, not necessarily the same, of course, by quite diverse pathogens. So future possibilities, well, immunisation against the virus in infancy, obviously very much in the future, but more feasible now because the age of primary infection is rising. Where it used to be infants, I mean all the people who looked at who are older would have been infected probably in infancy, but now people are getting, with rising socio-economic situation, the age of primary infection is rising. And so there might be more possibility of immunisation, which hopefully might stop the disease as well as cause of a very rare disease, herpes, simplex encephalitis, and use of antiviral agents to retard disease progression. And obviously, if this is done early enough in the disease, this would really stop a lot of deterioration. And just to show members of my very small group, these are people who've worked on this now or in the past, and a large number of collaborators who provided us either with specimens or advice. I think I'll finish there.
Thank you very much, Dr. Saki.